GMP manufacturing facilities located. We have subsidiaries in 28 countries and distribute to an additional of 73 countries. And currently, we employ over 3,000 employees worldwide. Our mission is to serve the needs in life science research and to translate new scientific findings into clinical applications. As for the agenda, first, we will have a brief look at the biology and function of T cells, as well as on the concept of workflows. Next, we will highlight our MAX technology and why it is the best for cell separation. After that, we will come to the main part of this presentation, the T cell separation product highlights. Finally, we will have a short summary, including a brief presentation of our highlight T cell workflows. Let's begin with the biology and function of T cells, as well as with the concept of workflows. Well, when talking about T cell phenotypes and function, mostly surface markers, cytokines, and transcription factors come into play. Accordingly, these will be most talked about when it comes to T cell separation, culture, and analysis. T cells play as effectors of the immune system an integral role in the adaptive immunity. They can be roughly subdivided into several phenotypically and functionally distinct subsets, such as T helper cells, cytotoxic T killer cells, T regulatory cells, as well as the rather heterogenic group of unconventional T cells comprising, for example, natural killer T cells and gamma delta T cells. T cells are the only immune cells that are able to form a long-lasting immunological memory that helps to respond faster and more efficient to already experienced threats. Key surface markers that are always mentioned in regards to T cells are the T cell receptor TCR that recognizes antigens, as well as the activating co-receptors CD3 and CD28. Those three surface markers are present on all T cells. Furthermore, co-receptors CD4 and CD8 are the defining markers for T helper cells and cytotoxic T killer cells, respectively. To exert their function as effectors of the immune system, T cells release cytokines to communicate with the environment and to cause various effects such as promoting inflammation. These include interleukin-2, interleukin-4, interferon gamma, tumor necrosis factor alpha, transforming growth factor beta, and interleukin-10. T cells are most relevant in both maintenance of health as well as fighting diseases. Last but not least, these properties make T cells ideal tools for immunotherapy, where T cells, for example, are genetically modified as CAR T cells to attack cancer specifically. Although our best-known products are, of course, the microbeads and isolation kits, we are not just a cell separation company. We actually cover the whole experimental T cell workflow from cell separation to cell analysis, including also sample preparation as well as cell culture. We provide thereby solutions for each step of your experiment. Today, however, in the first part of our T cell webinar series, we will indeed talk about T cell separation, including microbeads, isolation kits, and of course, automation. T cell culture and T cell analysis webinars will follow soon. We thought maybe the best way to arrange our quite large T cell portfolio are distinct workflows, specifically designed for your experimental needs. Thereby, we cover as focus workflows, amongst others, regulatory T cells, CAR T cell research, gamma delta T cells, tumor infiltrating leukocytes, polarization of T cells, antigen and virus-specific T cells, and of course, activation and expansion of T cells. With these comprehensive workflows, you can easily choose the product relevant for your specific field of interest. Please note that our T cell portfolio is growing by the day, and the respective workflows are all constantly evolving, so check regularly for updates. By the way, you may notice throughout the presentation small yellow buttons paired with a little click hand symbol, just the one here, like the one here on the lower right. In the PDF version of this talk, you can access the respective product pages, marketing materials, videos, and protocols by simply clicking these buttons, as they are all hyperlinked. Well, getting your whole workflow from Miltenyi Biotec means ease of mind. standardization, reproducibility, and organization of an experimental workflow become much more feasible. Miltenyi, as supplier for the whole T-cell workflow, means that all reagents fulfill the same high-quality criteria and the order process can be streamlined, thereby harmonizing the steps of an experiment as well as automation and upscaling are easily performed. Getting all products to Miltenyi guarantees smooth procedures and less errors, since, for example, our cell separation is tested for compatibility with our flow cytometry and our media for compatibility with our cytokines and so forth. Moreover, having Miltenyi as full supplier for the whole workflow means also getting full support for every step of your experiment, thanks to our great scientific support team. Moreover, a hallmark feature of Miltenyi's workflow solutions is the seamless translation from a research setting into a clinical setting and back, thanks to our Max GMP portfolio as well as cell manufacturing platforms such as the Clinimax Prodigy. Well, let's go to the next part of this presentation. Why Max technology is the best to isolate your T-cells. Cell separation poses many challenges to the researcher. High purity and recovery are of course important, but also is reproducibility, cell viability, and compatibility to downstream applications. Furthermore, there is a variety of different T-cell subtypes that might be of interest and have to be separated individually. Some of them display very low frequencies in the variety of different starting materials and are really hard to obtain. And of course, cell separation should be fast and easy to use. Consequently, there are many cell separation challenges to face, and gladly Miltenyi Biotec offers the right solution for all of them. But why use Miltenyi Biotec for cell enrichment, and what does Max technology make so special? Well, in short, it's our select the best approach. Minimal labeling with no effects on cell biology for full downstream compatibility. In more detail, our Max columns are a technological advantage, as the column amplifies the magnetic field by 10,000-fold. This allows for minimal labeling and smallest bead size of only around 50 nanometers. In consequence, less epitopes are covered on the cell surface and less strength is applied on the cells. So, as thousands of publications prove, the isolated T-cells are fully suited for any downstream application, including flow analysis. Thereby, Max microbeads were even approved by the FDA to be used in a specific clinical setting. It is important to note that the column allows for the most gentle sorting, since there is a large space between the steel spheres of the column matrix, with about 20 times the size of a lymphocyte. The T-cells will pass through smoothly without any negative effects. As you can see here, this is a small lymphocyte and there's really more than enough space between the steel spheres and the column. Have a look at the data on the right side with the CD3 labeled T-cells. Column-free technology results in bead aggregates in culture as well as in a clogged cell surface. With Max column technology, you see no bead aggregates in culture and minimal labeling of fully functional cells. And indeed, you see also in these Western blots that T-cells are not activated after cell isolation with Max technology. CD4 positive T-cells were isolated with either positive selection or negative selection using Max technology or a column-free competitor method. Their activation status was then assessed by analyzing the different isolated T-cell samples and performing a Western blot for MAP kinase phosphorylation, which is a reliable marker for T-cell activation status. On the left side, you see a Western blot for P38 phosphorylation after positive isolation of CD4 positive T-cells using either CD4 Miltenyi MicroBeads, human, or two column-free competitors. Untreated and LPS-treated SUPT1 cells served as negative and positive control, respectively. It can be clearly seen that cells isolated with Max technology yield much lower phospho-p38 expression than cells isolated with the competitor technology. On the right side, you see a Western blot for junk 1 and 2 phosphorylation after negative isolation of CD4 positive T cells using either Miltenyi CD4 T cell isolation kit human or 3 competitor column-free systems. Also here, untreated and LPS-treated SUPT1 cells served as negative and positive control respectively. Again, it can be clearly seen that cells isolated with MUX technology yield a much lower phospho-junk kinase 1 and 2 expression than the cells isolated with the competitor technology. Both Western blots clearly show that cell separation with MUX technology is the most gentle and does not activate T cells, regardless of positive or negative selection. Talking about positive or negative selection, generally MUX cell separation technology offers 2 different separation strategies. First, positive selection with our microbeads and microbead kits. Target cells are hereby labeled and directly selected via their specific and defining marker. As target cells are still magnetically labeled afterwards, they are usually not suited for an additional round of sub-separation, except when you use REAlease MicroBeads, where the label can be removed via an additional step. We will come later to this exciting technology. The positive selection approach with microbeads is very specific and ideally suited for rare cells or undefined single cell suspensions, such as tumor, as well as other difficult or compromised samples, for example, from sick donors. Also, isolation directly from whole blood and blood products is very well achieved with MUX positive selection. Secondly, we have the untouched isolation with our isolation kits, working via the depletion of non-target cells. This is necessary when the target cell fraction is not defined by a certain marker, but rather the lack thereof. Hereby, all cells expressing a defined exclusion marker cocktail are depleted, and the target cells remain unlabeled and thus are usable for further rounds of sub-separation. However, in order to work efficiently, the single cell suspension has to be rather well defined, as it is the case with, for example, human PBMCs or mouse spleen single cell suspensions. Taken together, different isolation strategies allow to get the most out of your sample, with Miltenyi being an expert in all cell separation strategies. Playing with and combining the different approaches offers a great flexibility according to your needs and is the secret to success. Consequently, MUX cell separation technology comes with 4 different approaches to perfectly reflect the different starting materials. First, we have microbeads and microbead kits that are ideal for positive T cell separation from PBMCs and various tissue single cell suspensions. They also include the new REAlease technology for label-free positive cell separation. Separation of basic T cells and almost any T cell subtypes from different species like human, mouse, rat, and non-human primate make microbeads and microbead kits a very versatile cell separation approach. Second, and almost equally versatile, we have the isolation kits that are ideal for all untouched separation needs. The used starting materials are PBMCs or defined single cell suspensions. Also, the isolation kits cover basic T cells as well as almost any T cell subtype from different species like human, mouse, rat, and non-human primate. However, if you do not want to get over a density gradient to your PBMCs as a starting material, you can now isolate T cells also fast and directly from whole blood and blood products. So as a third option, we have our StraightFrom MicroBead Kits for the isolation of human, CD3, CD4, and CD8 T cells directly from whole blood and blood products without the need for density gradient. It is a reliable way to get your T cells fast and easy, and is of course fully automatable. As fourth separation variant, we offer MUX Express isolation kits, Also here, human Tregs, pan-T cells, as well as CD4 and CD8 subsets, and even CD4 memory T cells can be isolated directly from whole blood and blood products without the need for density gradient. This time, however, it is an untouched cell isolation approach. Max Express is the fastest and most uncomplicated manual cell separation approach. One of the many things that sets Miltenyi Biotec's T-cell portfolio apart is its broadness and diversity. We cover more than just the basic human and mouse T-cell subsets. Indeed, over 90 T-cell separation products cover virtually every T-cell subset addressing T-cell researchers' needs worldwide. For example, different naive and memory T-cell subtypes as well as T helper cell subtypes are covered. Furthermore, different T regulatory cells, gamma delta T cells, and NK T cell separation kits are available, as well as complete workflows for antigen-specific T cells, cytokine secreting T cells, and CAR T cell research, and so many more. Apart from human and mouse samples, we offer also T cell separation kits for rat and non-human primate samples. As scientists, we understand how important it is to address the full complexity of T cell subtypes, even for the rare ones, and to offer according solutions. In the following slides, I will give an overview of the most important T cell separation products, including our highlights. So let's go to the main part of this presentation, our T cell highlight products. The first highlights of cell separation products in the T cell area are our T cell isolation kits for the untouched isolation of various human and mouse T cell subsets. Of course, the kits offer our well-known high purity in recovery. However, also a very important factor is, of course, the protocol speed. Especially with untouched isolation, faster protocol times are favored. Miltenyi Biotec offers therefore a total of 17 fast cell isolation kits that allow to isolate publication quality target cells in only 18 minutes. The 6 mouse fast isolation kits comprise the most important T cell subsets as well as 2 B cell kits. The 11 human fast isolation kits also cover the most important T cell subsets as well as B cells, NK cells, and monocytes. So instead of long and tedious processing, we offer a fast and easy protocol. However, it doesn't stop here with the isolation kits. Also, our best-selling mouse microbeads were improved and the protocols updated by reducing tedious washing steps and optimizing incubation times. In 3 easy steps, preparation, labeling, and separation, you get from spleen to target cells in less than 15 minutes. So it's simply straightforward. CD4 positive and CD8 positive mouse T cells, as well as CD90.1 mouse and rat T cells, and also CD90.2 mouse T cells can now be isolated via positive selection in only 30.5 minutes using our updated MicroBeads mouse. Of course, with the ever appreciated quality and purity. It is an easy and fast protocol that can be fully automated on the Automax Pro or the Multimax. Each microbead kit has a high capacity of 2 × 10⁹ total cells, which corresponds to up to 20 mouse spleens. Consequently, with their high sample capacity and automation options, the updated microbeads mouse are not only economic, but also very efficient and therefore ideally suited for the processing of high sample numbers. And again, it doesn't matter if you use positive or negative selection. Cell separation with MAX technology is fully downstream compatible and has no effects on the cell biology in either case. In this example, T cells were either positively selected with CD4 or CD8 microbeads, mouse, or isolated untouched with our own respective T cell isolation kits, which are the gold standard. Directly after isolation, here labeled with day 0, and after 1 day of cultivation, with and without stimulus using the T cell activation and expansion kit, marked with plus and minus respectively, activation markers CD25 and CD69 were analyzed by flow cytometry to assess a potential activation. As you can see, with without stimulation, cells are neither activated when separated positively nor when separated negatively with microbeads or isolation kits, respectively. This means that both cell isolation methods are equally gentle. When stimulated on purpose with the T-cell activation expansion kit, cells are highly activated and upregulate accordingly CD25 and CD69, regardless of the isolation strategy, meaning that both isolation methods are equally suited for functional downstream application. This is true for both CD4 and CD8 cells, as here shown in this example. Consequently, both approaches do not affect cell biology and ensure full T-cell functionality, while ensuring fast and gentle isolation. As a useful tip, which you see here in the box on the lower right, and these boxes are at several points in this presentation, we would like to suggest to not use red blood cell lysis, because this indeed activates your T cell before the isolation. However, the red blood cell lysis is not needed for T cell isolation with our Max microbeads from mouse spleen, so better avoid it completely. Our next highlight products are the StraightFrom MicroBeads. StraightFrom MicroBeads allow the isolation of target cells directly from blood and blood products without density gradient centrifugation. In regards to T cells, CD3 positive, CD4 positive, and CD8 positive cells are covered and can be directly isolated from whole blood, buffy-coat, leukoreduction system chambers, also called LRSCs, and leukopecs. Of course, cell separation with StraightFrom is automatable, depending on the starting material. This means processing on either Automax Pro or Multimax are possible and reduces processing time, for example, of a buffy coat to only 30 minutes. Indeed, protocols need to be fast and easy, especially for larger screening purposes. Automation and high throughput options may be desirable. Furthermore, starting materials, in the case of blood products, come in different formats, and restriction here is not desirable. Moreover, sometimes samples and cell numbers are limited. So, of course, cell loss, especially when working with small or rare samples, must be minimized and purity maximized. However, most importantly, cells must not be activated by the isolation to ensure that, for example, really only the specifically stimulated T cells proliferate and are fully functional. StraightFrom is here the ideal solution, a fast and flexible positive isolation approach that allows to isolate different cell types from the same sample with minimal loss due to the skipping of density gradient centrifugation. This makes StraightFrom technology also ideal for the isolation of T cells, for example, in the field of CAR T cell research. To date, the most widely used method to isolate target cells from whole blood and blood products is, however, performing a density gradient centrifugation for the generation of PBMCs, followed then by cell separation of the target cell subtypes. Apart from the time-consuming protocol with over 2 hours processing time, the handling is rather complicated and does not allow for automation. Furthermore, a significant amount of target cells may be easily lost in a user-dependent fashion. In addition, harsh and long handling as well as some gradient media may affect the cells, at least to some degree, so act carefully. The alternative to density gradient centrifugation is Miltenyi's StraightFrom technology. Hereby, cells can be directly isolated from blood products in a fast and easy way, without density gradient centrifugation, cell counting, or washing. The procedure is fast but gentle and can be fully automated. So, from sample to target cells in only 30 minutes. Fast, easy, and convenient. This is, of course, also backed up by flow cytometry data and the assessment of activation marker expression after isolation. In this example, CD4 positive and CD8 positive T cells were isolated from buffy coat, either with StraightFrom CD4 CD8 microbeads on the left side, or via density gradient centrifugation to get the PBMCs followed by a competitor's untouched pan-T cell isolation, like here on the right side. Cells were then activated for activation marker expression of CD25 and CD69, either directly after cell isolation, here labeled with day 0, or after 1 or 2 days of cultivation, either without stimulus or stimulated with the T cell activation and expansion kit, labeled with minus or plus, respectively. Cells isolated with StraightFrom show almost no activation marker expression even after 2 days of culture. However, when activated on purpose, using the T cell activation and expansion kits, the cells highly upregulate CD25 and CD69, showing their full biological functionality. Cells that were isolated by density gradient centrifugation, followed by a competitor's untouched pan-T cell isolation, instead showed higher activation marker expression from the beginning on directly after isolation and 1 or 2 days later, and consequently may not be efficiently activated in downstream applications such as polyclonal or antigen-specific activation. Our next T cell separation highlights are the REAlease MicroBead Kits. This revolutionary microbead technology allows to positively select cells, but then remove the microbead and the antibody labeling to get completely label-free cells. REAlease MicroBead Kits are available for human and mouse T cell and non-T cell subtypes, including markers such as CD3, CD4, CD8, CD19, CD25, CD56, CD62L, CD90.1 and 2, CD279, also called PD1, and TCR gamma delta. This offers researchers an extremely and entirely new level of flexibility and new separation strategies combining the best of 2 worlds, highly specific positive selection and, if needed, label-free, for example, for further subset separations. The procedure itself is very easy. After cell separation, there is a bead removal step with the bead release reagent. Cells are now ready for a second round of magnetic labeling. And an additional optional step can then be applied using the REAlease reagent to also remove the bound antibody biotin complex from the cell surface, thus to obtain completely label-free cells and make all epitopes accessible again. However, the advantages of REAlease do not end here. REAlease is so gentle and minimally invasive due to the special structure of the REAlease complex that even cell isolation via CD3 does not activate the T cells, regardless of the label removal. As you can see in the example here of isolated human CD3 positive T cells on the left side, activation marker CD69 is not upregulated amongst pre-gated CD4 positive T cells, neither after CD3 labeling, nor after isolation, nor after label release. However, when activating cells on purpose, CD69 is nicely upregulated and shows the biological function of the T cells. Please note that pre-gating on CD8 positive T cells showed exactly the same results. The example on the right side, however, shows a multi-step subset isolation using REAlease. First, CD4 positive T cells are pre-selected. Then the label is removed and cells are now ready for a second round of deeper subset separation, in this case via CD25 for Treg isolation. With that, you can actually get your Treg from almost every single cell suspension. Further examples that are similar include, for example, the isolation of gamma delta T cells as well as CD3 negative NK cells. Another fantastic and very specialized cell separation product are the TIL MicroBeads. The microbeads have a special formulation to facilitate the isolation of tumor infiltrating leukocytes, abbreviated TILs, from tumor single-cell suspensions. TILs are very rare cells, and the tumor tissue is a heterogenic and hard-to-process starting material. This poses quite a challenge to the researcher and the successful isolation of TILs. In this context, the specialized TIL microbeads are the ideal tool to efficiently enrich viable and pure TILs from tumor single-cell suspensions with highest recovery rates. The TIL microbeads are available for human and mouse CD45, CD3, CD4, and CD8 positive cells. The human TIL microbeads are moreover available as REAlease version to facilitate label-free isolation of target cells from human tumor tissue. The TIL microbeads are best integrated in our wholesome and comprehensive TIL workflow. So ideally combine our TIL isolation solutions with our dedicated sample preparation solutions, flow portfolio, and imaging portfolio for best results. Please have also a look at the respective application and product pages as well as the webinars by following here the links on the right side. Finally, I want to point out a cell separation product that is absolutely unique, the cytokine secretion assays. They allow to detect and enrich viable cytokine secreting cells from mouse and human samples. Thereby, cytokines are immobilized on the surface of the secreting cell by the so-called catch reagent. Basically, thereby, the cell labels itself with its own cytokines. In a second step, the immobilized cytokines are then labeled with fluorophore conjugated antibodies called detection reagent and can then be detected viably without intracellular staining on any flow cytometer. In an optional third step, the fluorophore-labeled cytokine-secreting cells can be specifically enriched and used in downstream assays by using anti-fluorophore magnetic beads. Consequently, our cytokine secretion assays come in two versions. For flow detection only, we have the detection kits. For the separation of viable cytokine secreting cells, we have the detection and enrichment kits, including the additional enrichment step via a MAX column. MAX technology offers, depending on the researcher's needs, different manual and automated cell separation options for upscaling. Manual separation with the Quattro or OctoMax for up to 4 or 8 samples in parallel, respectively. Automated options include the AutoMax Pro for full walk-away convenience for up to 5 samples, the Multimax Cell 24 Separator Plus for the processing of up to 24 samples in parallel. And finally, the Multimax X for flexible, fully automated, high-throughput industrial applications with any source material. Now, every researcher can reliably automate cell separation for more efficient workflows and higher consistency. Of course, all the automated options are applicable to most of our T-cell separation solutions. Finally, we can start to sum up this talk and briefly present some of our key T-cell workflows. As mentioned before, the variety of our T-cell products is best organized in comprehensive workflows following your scientific interest. Consequently, please find here listed our highlight application workflows with clickable links to the respective application and protocol pages, as well as to further info material and webinars. Of course, there are more, and our selection is growing by the day. However, our highlights include polarization of T cells with protocols and cytokine combinations for the most efficient polarization of your CD4 positive T helper cells. The regulatory T cell workflow with all needed solutions for the isolation as well as the expansion and analysis of rare Tregs in a translational setting. The CAR T cell research workflow covering all steps from T cell isolation to activation to transduction, and finally CAR T potency analysis for fully translational and functional generation of CAR T cells in a research or small-scale setting. The TIL workflow to efficiently dissociate tumor tissue and isolate even the rarest tumor-infiltrating leukocyte. The antigen-specific T cell workflow optimized for the specific stimulation of antigen-reactive T cells, as well as the isolation and analysis of these extremely rare cells in different contexts. And finally, the COVID-19 virus-specific T-cell workflow that covers each step from stimulation to enrichment to analysis of SARS-CoV-2 reactive T-cells. Have a look at the respective application pages and webinars to find out more. Although our T-cell portfolio is quite extensive, as you can see here in the full T-cell workflow overview, you have to remember just 3 easy things. We offer the best T-cell separation, but we cover also the whole T-cell workflow. And finally, we have a solution for every pain point. So have all the success with our innovative and complete T-cell portfolio as displayed here in this full-featured overview. Our T-cell portfolio is growing by the day, and the respective workflow overviews are constantly evolving, so check regularly for updates. Please follow also the links on the lower right to access the respective T-cell product pages as well as to download our T-cell brochures. Talking about more information, of course you can find a variety of helpful marketing material downloadable from our webpage www.miltenyibiotec.com for T-cell specific applications. Just click on the button on the lower right side. The information includes flyers and brochures, webinars and videos, protocols and data sheets, customer reports, application notes, scientific posters, and data sets. And of course, you can always directly reach out to the responsible sales representative in your area. I hereby end this presentation with this nice picture of our company campus in Bergisch Gladbach. Thank you very much for your attention.